So let's move along to the human papilloma vaccine session, please, and Dr. Bocchini.
Thank you, John. First, I want to just add my thanks to Dr. Pickering for his years of leadership and his accomplishments as the Secretariat of ACIP. We've shortened the presentation for HPV based on the time limits and are trying to provide you with the essential information which would lead to a vote. As you know, the 9-valent HPV vaccine was licensed by the FDA in December. It was licensed for females 9 through 26 years of age and males 9 through 15 years of age, and trials were conducted with the 3-dose schedule. This is an L1 viral-like particle vaccine similar to the quadrivalent HPV vaccine, which targets 5 additional high-risk oncogenic types, which are listed here. In the United States, these are estimated to cause 14% of HPV-related cancers in women and approximately 5% of HPV-related cancer in males. Males 16 to 26 years of age were not part of the BLA submitted in 2013. We did see data from the trial on 16 to 26-year-old males for vaccine safety and seroconversion rates. This was presented to ACIP in October, and now a supplemental BLA has been submitted for this age group to the FDA. So, as you're aware, the current recommendations are for routine vaccination at age 11 or 12 years of age. Vaccine can be given as early as 9. Vaccine is recommended through age 26 for females and through age 21 for males who have not been previously vaccinated or who have not completed the series. And vaccination is recommended for immunocompromised persons, including persons who are HIV infected, and for men who have sex with men through age 26. Those are male recommendations, and it's a 3-dose schedule given at 0, 1 to 2, and 6 months of age. So the 9-valent vaccine, going backwards. So now we have 3 available HPV vaccines, which you're familiar with. The 9-valent is added to the list of licensed vaccines. In preparation for this vote, we have provided a number of presentations over the past year, and including in October a review of the grade for the 9-valent vaccine, a health economics analysis, and we discussed policy options for which we got significant feedback from ACIP members. Today, actually, these presentations have been modified. Dr. Harold Chesson will provide some information on vaccine impact and cost effectiveness. Dr. Markowitz will then provide considerations for recommendations, which will lead to ACIP discussion and vote, and then a VFC resolution will follow. I want to thank the work group members. We've had considerable number of discussions and calls over the past number of months, and especially recognize Lori Markowitz for her leadership as the ACIP CDC lead.
Good morning, or good afternoon, I should say. So I'm Emika Petrosky. I'm an EIS officer who's been working with Lori Markowitz, and I'd like to also thank the work group as well. We've worked on this quite a bit this year. I was advised to speak slowly. So this is an outline of my presentation. In the interest of time, I'm going to keep this very brief. Listed here are the 9-valent vaccine studies, which have all been previously presented to the ACIP. As a reminder, Protocol 1 is the pivotal efficacy study that compared the 9-valent vaccine to the quadrivalent vaccine in adult females. There were also immunobridging studies and concomitant use studies, and one study assessing the 9-valent vaccine in prior quadrivalent vaccine recipients. I'm only going to present the data from Protocol 1, as this was the pivotal efficacy study, and will then summarize the results and safety findings from the other studies. For the 5 additional types, the 9-valent vaccine demonstrated greater than 96% efficacy in preventing high-grade cervical, vulvar, and vaginal disease and 6-month persistent infection. For outcomes due to the 4 original types, the incidence of disease in either vaccine group was very low, and the 9-valent vaccine demonstrated comparable protection in preventing high-grade cervical disease and anal genital warts. Both the 9-valent vaccine and quadrivalent vaccine induced greater than 99% seroconversion, and the non-inferiority criterion was met for the 4 original types, as shown here. I'll now summarize the findings from the 9-valent vaccine studies. As I just showed, the 9-valent vaccine demonstrated close to 97% protection against outcomes due to the 5 additional types, and similar protection against disease due to the 4 original types. There was very few incidents of disease in both the 9-valent and quadrivalent vaccine cohorts. The 9-valent vaccine also demonstrated non-inferior immunogenicity compared to the quadrivalent vaccine for the 4 original types. Although I did not show this data, the vaccine also demonstrated non-inferior immunogenicity for all 9 HPV vaccine types in adolescent females and males compared to adult females, and in adult males compared to adult females, supporting the bridging of efficacy findings in adult females to these other groups. Finally, the concomitant use studies demonstrated that the 9-valent vaccine can be administered concomitantly with the other adolescent vaccines listed here. Regarding safety, the 9-valent vaccine was generally well tolerated in over 15,000 recipients and had an adverse event profile similar to the quadrivalent vaccine across age, gender, race, and ethnicity, with the exception of a higher frequency of injection site swelling and erythema in females. Males had a lower frequency of adverse events compared to females, which is similar to what was seen in the quadrivalent vaccine program. I'll now review grade of the 9-valent vaccine that was presented to the ACIP last October. As a reminder, the policy questions developed by the work group were: should the 9-valent vaccine be recommended routinely for 11 or 12-year-olds? And should the 9-valent vaccine be recommended for females aged 13 through 26 years and males aged 13 through 21 years who have not been previously vaccinated, which I will refer to as catch-up vaccination. This slide shows the grade summary table for routine 9-valent vaccination in females. I will not go over this table in detail, as this was already presented to the ACIP. If you'll recall, the work group gave the overall evidence type for routine vaccination in females a ranking of 2, indicating that the data reflected a moderate level of evidence. For catch-up vaccination in females, the work group also gave this a ranking of 2. For routine vaccination in males, the work group gave this an overall evidence type ranking of 3, indicating a low level of evidence. And this was the same for catch-up vaccination in males. I'll now present the considerations for formulating 9-valent vaccine recommendation, several of which I just reviewed. Regarding the evidence type for benefits and harms, the data for the 9-valent vaccine are from randomized controlled trials and immunobridging studies. The work group gave an overall evidence type of 2 for females and 3 for males. The work group felt that the benefits of 9-valent vaccination outweigh the harms and placed a high value on the prevention of outcomes due to the 9 HPV vaccine types. The 9-valent vaccine is expected to be cost-saving compared to the quadrivalent vaccine. In the next presentation, Dr. Chesson will provide more information regarding cost-effectiveness. In conclusion, the work group proposes a Category A recommendation for the 9-valent vaccine. Dr. Markowitz will be going over the specific recommendations for the vaccine during her presentation today. Thank you.
Thank you very much for a very nice, succinct presentation. Dr. Chesson.
Good afternoon. Today I will summarize the 3 models we have available of 9-valent vaccination in the United States, and then I will summarize the unpublished results regarding the impact and cost effectiveness of 9-valent vaccination. At the October meeting, Dr. Brisson presented the US HPV-ADVISE model, which is based on a published 18-type Canadian model, which has been fitted to US data. The Merck model and our simplified model are both based on published 4-valent versions of the model, and these models have been expanded to include the additional types in the 9-valent vaccine. All three of these models are dynamic, meaning that herd effects are included in the results that I'll be showing, and all of the models include a wide range of health outcomes that can potentially be averted through vaccination, including cervical cancer and other HPV-associated cancers. In the interest of time, I won't go over all of these model characteristics, and I won't throw modeling jargon at you, but I'll just point to the second row here and say that in terms of the degree of complexity and comprehensiveness, the HPV-ADVISE model ranks highest, our simplified model lowest, and the Merck model falls in between the two. And I won't go over all these model assumptions as well, but I will point out that all the models assumed a high vaccine efficacy for both the 9-valent and the 4-valent vaccines and assumed a lifetime duration of protection. And all of the models use the same cost assumptions shown here, such that the 9-valent vaccine was assumed to cost $13 more per dose than the 4-valent vaccine. So now I'll move on to the impact and cost effectiveness results. But before showing those results, I just wanted to point out in absolute terms the potential for additional cancer prevention in the U.S. This is a slide from Dr. Person's presentation. And as you can see, the majority of the benefit of the vaccines shown in gray from preventing 16 and 18 from the quadrivalent vaccine. And then in blue, we see the additional reduction. So the greatest gains from the 9-valent vaccine are in terms of cervical cancer prevention. This graph shows the estimated effectiveness of the 4-valent and 9-valent vaccines in a scenario in which no cross-protection is assumed for the 4-valent vaccine. The red line shows the scenario in which the 4-valent vaccine is provided to boys and girls, and the blue line shows the scenario in which 9-valent vaccine is provided to boys and girls. So for cervical cancer, for example, in the long term, there's a 65% reduction with the 4-valent vaccine. And if both sexes were switched to the 9-valent vaccine, there'd be an additional 14 percentage point decrease in cervical cancer. Again, this is for the no cross-protection scenario. If we did assume cross-protection for the 4-valent vaccine would have a bigger impact, and the marginal impact of the 9-valent vaccine would be reduced slightly. This graph looks at sex-specific 9-valent vaccine strategies. And again, the two outcomes are CIN2-3 and cervical cancer. And the red line again shows the scenario in which the 4-valent vaccine is provided to both sexes. The dotted blue line shows the scenario in which girls are given the 9-valent vaccine and boys are given the 4-valent vaccine. And then the solid blue line shows the scenario in which girls and boys both get the 9-valent vaccine. So for these outcomes, the majority of the benefit of switching to 9-valent vaccination is achieved through switching females to the 9-valent vaccine. This table shows the cost-effectiveness results from the US HPV-ADVISE model under the scenario of no cross-protection for 4-valent HPV vaccine. The top row compares a strategy of 9-valent vaccination for females and 4-valent for males to a strategy of 4-valent vaccine for both sexes. So what this comparison tells us is what is the incremental cost effectiveness of switching females from the 4-valent to the 9-valent. And the model indicates that this is cost saving, indicating that the additional cost of the 9-valent are offset by the averted medical costs due to the additional benefits of the 9-valent vaccine. The next row shows the comparison of a strategy of 9-valent vaccine for both sexes to the strategy of 9-valent vaccine for females and 4-valent vaccine for males. What this tells us is the incremental cost effectiveness of switching males from 4-valent to 9-valent, and the model came in at $31,000 per quality adjusted life year. The bottom row shows us the scenario of comparing 9-valent vaccination for both sexes to 4-valent vaccination for both sexes. So what this comparison tells us is what if we simultaneously switch both sexes from the 4-valent to the 9-valent, and the model suggests that the cost per quality is less than zero or cost saving. And we think this bottom row is the most applicable to the decision that we're facing now, because sex-specific vaccine strategies are likely not a viable policy alternative in the U.S. So this is the bottom line result from the US HPV-ADVISE model. So I'll now show you the bottom line results from all three of the models. So this summarizes the cost effectiveness of 9-valent vaccination for both sexes to 4-valent compared to 4-valent vaccination for both sexes. The first column of results shows the scenario in which there's no cross-protection assumed for the 4-valent vaccine, and all three models indicate that this strategy is likely cost-saving. When we assume cross-protection for the 4-valent vaccine, the results are still similar across models in that the cost per quality gained by the 9-valent vaccination is low. It remains cost-saving in the HPV-ADVISE model, and it is only $8,000 in the simplified model. The results were consistent in a range of sensitivity analyses when we compared 9-valent vaccine for both sexes to 4-valent for both sexes. The mean cost per quality gained was less than $0 in most every scenario we examined. There were a couple of exceptions when we assumed cross-protection for the 4-valent vaccine, as shown here. But I want to point out that at the uncertainty intervals that were generated in these sensitivity analyses, the cost per quality gained by the 9-valent vaccine remained less than $10,000 when assuming no cross-protection for the 4-valent, and remained less than $25,000 when assuming cross-protection for the 4-valent vaccine. So in conclusion, the current 4-valent vaccine program is expected to reduce HPV-related diseases substantially in the United States, and switching to a 9-valent program is expected to further reduce precancerous lesions and cervical cancer. The HPV-ADVISE model suggested reductions of 19% and 14% in CIN2-3 and cervical cancer, respectively, with perhaps more modest reductions in the other HPV-associated cancers. Providing 9-valent vaccine for girls provides the great majority of benefits of providing 9-valent vaccination to both sexes. And to conclude, for the cost effectiveness, primary 9-valent vaccine for both sexes is likely cost saving compared to 4-valent vaccine for both sexes. And again, these results are consistent not only across the three models, but within each model as we varied the assumptions. The cost per quality did not exceed $25,000 in the sensitivity analyses. It was less than $0 in most of the scenarios we examined. And finally, analyses of additional 3-dose 9-valent vaccination for prior 3-dose 4-valent vaccinees is underway, and preliminary results are highly variable because the incremental health benefit in terms of the gain per person vaccinated is quite small. Thank you.
Thank you, Dr. Chesson. And Dr. Markowitz.
Okay, so what I'm going to do is review the proposed HPV vaccination recommendations that have been discussed by the work group. And I want to mention just from the start here of my presentation that we will not be addressing the last point that Dr. Chesson mentioned, which is the providing 9-valent vaccine to individuals who've already received quadrivalent vaccine. This is an ongoing consideration. We're doing additional modeling, and in light of the time restrictions of the ACIP, at this time, we didn't feel we had time to address that adequately in the session. So after we get approval for these, we are planning on publishing a MMWR policy note. And what I'd like to do today is go over specific sections of the recommendations, which include routine recommendations, some changes in the section on administration and intervals, interchangeability. I just want to give a brief update on the pregnancy registry issues and then discuss a section called future policy issues. So for routine recommendations and age groups, the work group proposes the same age groups as in our current recommendations. For these recommendations, 9-valent vaccine use in males older than age 15 would be off-label at present. And as discussed previously in this session, immunogenicity on males 16 to 26 were presented to ACIP in October, included in GRADE, and have been submitted to FDA. Compared with quadrivalent vaccine, 9-valent vaccine would provide a little additional benefit for males. However, programmatic issues were considered for these recommendations, including the low likelihood that providers would stock 2 different vaccines, and then the fact that eventually there will be a transition from quadrivalent to 9-valent vaccine. And this is the proposed recommendation. On this and subsequent slides, the underline indicates the new wording. And I'm not going to read the beginning of this because it's exactly the same as the recommendation that Dr. Bocchini went over in the introduction in terms of the age groups for males and females. The new section will be vaccination of females as recommended with bivalent, quadrivalent as long as this formulation is available, or 9-valent vaccine, and vaccination of males is recommended with quadrivalent as long as this formulation is available, or 9-valent vaccine. And there'll be a link to the grade tables at this point in the recommendations. This slide shows some additional wording for the routine recommendations, and I want to point out that the proposed wording does not specifically state a preference for any vaccine, but outlines the differences between the vaccines and what they can protect against. Now, the next session for discussion is on administration and intervals. In our current recommendations, the wording in this section is the second dose should be administered 1 to 2 months after the first, and the third dose 6 months after the first. Of note, studies of bivalent and quadrivalent vaccines show that longer intervals between doses do not result in lower antibody titers, and some studies have found higher titers after longer intervals. The workgroup discussed the pros and cons of mentioning flexibility in intervals. Some workgroup members felt this was important, and others had concerns about possible confusion for providers. And of note, the schedule on the intervals on the schedule will remain the same of 0, 1 to 2, and 6. But this is the wording that has been proposed. Bivalent, quadrivalent, and 9-valent are each administered in a 3-dose schedule. The second dose is administered at least 1 to 2 months after the first dose, and the third dose at least 6 months after the first dose. Some studies found higher antibody titers after longer intervals between doses. And if the vaccine schedule is interrupted, the vaccination series does not need to be restarted. The next section I'd like to discuss is on interchangeability. There's been no study of interchangeability of HPV vaccines. However, there are some data on the 9-valent vaccine after 3 doses of quadrivalent vaccine from a study that was conducted as part of the 9-valent clinical program. The workgroup discussed programmatic issues related to transition to the 9-valent vaccine for consideration of wording changes in this section. And this slide shows the proposed wording. Again, the crossouts and the underlines recommend things to remove and add. We have deleted the first sentence in our current recommendations, which states that ACIP recommends that the vaccination series for females be completed with the same vaccine product whenever possible. And instead, we propose starting with the second current sentence, which says, if vaccination providers do not know or have available the vaccine product previously administered, or are in settings transitioning to 9-valent vaccine, for protection against 16 and 18, any HPV vaccine product may be used to continue or complete the series for females. And quadrivalent or 9-valent vaccine may be used to continue or complete the series for males. There's no change in our recommendations for vaccination during pregnancy, and I'm only showing this as an update. Information on vaccine and pregnancy registries have been changed a bit. There will be a pregnancy registry for 9-valent vaccine, and of note, earlier this month, the bivalent vaccine registry was closed with concurrence from FDA. So we will add wording about the 9-valent vaccine and make a note that both the quadrivalent and the bivalent vaccine registries have been closed, and there'll be links to phone numbers. The quadrivalent vaccine was closed previously. And finally, the short section on future policy issues is proposed at the end of the policy note. The workgroup discussion here centered on what should be included in the policy note related to an ongoing trial of 2-dose schedules for 9-valent HPV vaccine. Some workgroup members were concerned about, again, confusion for providers leading to possible delay in vaccination. However, we came up with a compromise, and this is the proposed wording for this very short section. A clinical trial is ongoing to assess alternate dosing schedules of 9-valent vaccine. ACIP will formally review the results as data become available. HPV vaccination should not be delayed pending availability of 9-valent vaccine or a future clinical trial data. I mentioned that we're not going to be considering this, but I want to let you know that the workgroup is discussing 9-valent 9-valent HPV vaccine for persons who completed a HPV vaccination series. Importantly, the manufacturer did not seek an indication for 9-valent HPV vaccination in persons who had previously received HPV vaccine. However, there was one study that was conducted, and the data are included in the label. I'm not going to review this today, but skipping down to the last bullet, as I mentioned, due to time limitations, this will be presented to ACIP at the next meeting. I want to thank all the work group again. I know this was mentioned earlier. We've had a lot of conference calls over the last few months and the last few weeks particularly. And again, for discussion and vote, just for approval of the routine recommendations and age groups that I reviewed, the wording on administration and intervals, interchangeability, and future policy issues.
Thank you, Dr. Markowitz. And I'm just appreciative of the entire HPV working group for a very nice, complete, and very succinct presentation that was very understandable. We're open for any questions or comments. Dr. Campos-Otko.
Yeah, can you put the slide back up with the suggested language again?
For the main one?
Yes.
Okay. There's two slides. One is, this is our basic recommendation for the age groups. And all we're doing at the end, which we had before, is mentioning which vaccines can be used for females and which vaccines can be used for males. But actually the basic recommendation, which is routine at 11 or 12, starting at age 9, 13 to 26 for females and 13 to 21 for males, has not been changed. We will have a footnote, by the way, that links to the recommendation for men who have sex with men and immunocompromised persons, and that's in a separate section in the policy note.
Okay, thank you.
And Ms. Pellegrini.
Thank you. First, I want to thank the work group very much for the revised language on administration. I think that's actually going to be very helpful in our parent education, saying that you don't have to complete the series within that very restrictive time frame for your teenager, but it can be spread out if necessary or if that's the way life goes. Could we look at the draft wording on interchangeability to slide, I think it's 9. If I'm reading this correctly, it seems like there's an implicit recommendation not to use bivalent. I mean, we're saying you should use quadrivalent or 9-valent to complete a series. So you can sort of go, you can give more strains, but you should not complete a series with bivalent if you don't know what it was started with. Is that correct?
Well, that's for males. For females, we basically are saying that any vaccine can be used to continue or complete the series. And this is the last time, since we only had one vaccine that was approved and recommended for males, we didn't have to have a separate statement for males. So that's why there's two separate statements now here.
Okay, maybe just for future discussion, I'm not sure why I'll communicate with the work group offline. Thank you.
Dr. Herman.
On that same language, when I read that, it sort of indicates to me that if you've got quadrivalent vaccine and that's what somebody started with, that's preferred. I mean, that's sort of how I would read that, perhaps if I were a provider. But I think you're saying that it doesn't matter, but yet the language sort of reads like if you can't give quadrivalent, then you can give the 9-valent. So I just kind of wonder about that language a little bit, unless you really do think that somebody should continue with the same vaccine, the quadrivalent. But
well, we've tried to soften the language by removing that first sentence, because I think that is what that first sentence did say, that ACIP recommends that the vaccination series for females should be completed with the same product whenever possible. And we wanted to liberalize this, acknowledging what was going to be happening during the transition. I think this is as far as we felt comfortable going. Dr. Bocchini, do you agree with that? Yeah.
so is there a preference to use quadrivalent if that's what was already started? If there's not, then it almost seems like you could use the language that's used for males.
I feel like we tried to make the language for males and females the same. They are the same. The only difference is the vaccines that are recommended for females and males. So maybe we need to have the same, is the part of the language that you think expresses that if vaccine providers do not know or do not have available or are in settings transitioning?
Right, it just seems like it could just read either if the vaccine series has already been started, either vaccine can be used to complete the series, rather than if you don't have it, or if, I don't know, maybe I'm the only one that would read it that way.
Well, and let me just say that we meant that sentence to apply to both the males and the females. We didn't mean it to read that was just for the females.
Dr. Bennett.
I don't know if this is the confusion, but for females, you could still be giving the bivalent vaccine. So I think that's what you were leaving room for, whereas for males, that would not be indicated. So.
And Dr. Markowitz, just for a reminder, can you give us an indication what percentage of HPV vaccine in the United States is a quadrivalent currently?
Yes, yeah. Probably over 98% of vaccine being used is quadrivalent. So our recommendations are really geared towards people who are getting quadrivalent vaccine.
So in essence, you're being very, very inclusive here, but also acknowledging what reality is. So I think that
That's a good point,
is conveyed nicely here. Yes, Dr. Sawyer.
In your language about extending the interval, the statement that some studies have demonstrated higher titers with longer intervals will invite providers to ask, well, how much longer? And how are you going to advise them in your policy statement about the specific data? Or are you just going to leave it open that longer intervals can be as long as you want?
We can add some clarification that's we have not because we have for a policy note we do not have a lot of room in the document. We have not gone into that in detail, so maybe we should add something. Is that your suggestion, or do you think we should take that out?
I think you need something. Otherwise, those of us who field questions from providers about intervals are going to get phone calls about, okay, well, should I wait 12 months? And what if I wait 15 months? And so we need something to give a boundary around that, or at least what the data shows.
Okay.
And then the provider can decide.
I think that's a good suggestion.
Dr. Lett.
Thank you. Laura, you indicated that you didn't have time today to talk about the data around revaccination, but will some guidance about that still appear in the policy note, even if you're not presenting it to the ACIP today? In vaccination of people who've already completed a series with, say, quadrivalent vaccine?
I think that's something that maybe we can ask the ACIP members how they feel about. We have not been able to discuss that, so we could be silent on that and just leave something out entirely, or we could indicate we could indicate someplace even in the future policy issues that we were talking about that, or we could have some type of statement in the policy note if the ACIP members feel that's important. This is an evolving issue based on the limited ACIP meeting that we're having.
Right.
Dr. Shuckett.
Yeah, if this issue needs the ACIP to deliberate, the ACIP has not been able to deliberate. There are other ways that CDC can make some factual information available to clinicians pending ACIP deliberating on this. So in terms of the evidence base that's out there and the not contraindicated and that kind of stuff. So I think we can manage some things for the confusion that may be out there pending letting ACIP thoughtfully deliberate on what they want to say.
And this will definitely come up at the June meeting, so I'm not uncomfortable with having a 4-month window here, especially when we're talking about an off-label use for a new product that people are just getting familiar with. So I think we're probably within our capacity right now. Dr. Harrison.
I'm a little unclear. Are we anticipating a vote in June or just an informational session in June about the use in previously vaccinated with the quadrivalent?
Well, I would assume that we would be anticipating a vote on the wording, because if people would want that included in a policy statement.
I'm just wondering whether we shouldn't consider an informational session before the next meeting for both this issue and for the meningococcal B issue. I mean, in the past, we've had some difficulty being presented with somewhat not straightforward information or decisions on the spot and then taking a vote during the same meeting.
Dr. Shuckett?
That's not going to be possible.
We can certainly provide some information to people as background materials before the meeting. I think that would probably be the best way to get the ACIP members, give them enough background information to be able to address this. So we can certainly do that as part of the regular process.
Oh, yes, Dr. Middleman.
Middleman from the Society for Adolescent Health and Medicine. I'm just looking at the wording that is up there and wondering if, given the open-endedness, as Dr. Sawyer had mentioned about the longer intervals, would it be helpful, do people think it would be helpful to add a statement about efficacy only being known if the series is completed? Or something along those lines to make sure there's completion as the end game?
Well, I mean, one other way that might avoid confusion would be to eliminate this sentence entirely that some studies have found. We can just say the second dose should be administered at least 1 to 2 months and the third dose at least 6 months, and we can eliminate this sentence, which seems to be causing some confusion. I think that would serve the same purpose.
I agree with that. Thank you.
I think that's a good idea as well, just to take the ambiguity until it's further discussed. Do we have other discussion or questions?
And I just want to mention about that, it's not that different from our current recommendation, which we say if the vaccination schedule is interrupted, it doesn't need to be restarted. So we've already made the statement, we're just clarifying it by highlighting the individual intervals.
Very good. So we have some draft wording up here that we've gone through. Is there any tender of a motion? Dr. Rubin.
I make a motion to accept it with the deletion of that sentence on the slide regarding the antibody levels being higher.
And is there a second? I think I saw Dr. Vasquez. Okay. Further discussion? And seeing none, we will start with Dr. Kemp and go to your left.
Can I clarify, we're voting on all of this, or are we just voting on this one?
No, we're going to vote on the entire package because I think we have gone through and made changes where we felt appropriate. So if you don't mind, we'll just...
Yes, that's good.
So...
question?
Yes, Dr. Kimberlin.
So that whole packet, David Kimberlin, AAP, that whole package that's being voted on includes use of HPV-9 in prior recipients of HPV-4? But that wording was presented.
The wording is such that a patient who, where we don't know what the previous vaccine was, one could use for males HPV-4 or HPV-9. For females that we don't know what they had received before, they could receive the bivalent, the quadrivalent, or the 9-valent for the protection of 18 and 19, or 16 and 19. 16 and
18.
18,
Yeah.
thank you.
But draft wording for use of HPV-9 in persons previously vaccinated with HPV-4, that is not what's being voted on.
Yeah, because that had been distributed before the compressed session when we knew we were not going to have enough time, so we had took that off the agenda.
Any further clarification? And seeing none, I'm going to go back to Dr. Kemp.
Kemp, yes.
Karen, yes.
Belongia, yes.
Harrison, yes.
Harriman, yes.
Romero, yes. Rubin, yes.
Bennett, yes.
Tempty, yes.
Pellegrini, yes.
Reingold, abstain.
Campus Outcall, yes.
Vasquez, yes.
Okini, yes.
Riley, yes.
And the motion passes with 14 votes for and 1 abstention. So thank you very much to the HPV work group. And my apology for not taking any formal breaks here. And I'll just remind people, feel free to get up as you need. And Dr. Freihofer has a comment.
Sandra Freihofer, liaison for ACP. I understand the time constraints of this current meeting, but just to put it out there, I think the question about revaccination with HPV-9 for patients that have received HPV-4 is certainly something that a lot of practitioners ask, and just really look forward to guidance from ACIP and getting the word out there as to what we should do. Thank you.
Thank you. So I think at this point, Dr. Santoli is coming up for the,
I'll make this very fast. Kathy Newsel, IDSA. I think one of the most common questions we'll get after these new recommendations are seen will relate to the timeline of the availability of the 9-valent and the supply. So it would be helpful if somebody before the end of the session could comment on those two issues.
Is that something you're able to?
I can say a little bit. I think I'll also defer to colleagues from Merck to speak.
And if Merck could come on up.
Hi, I'm Julie McCafferty with Merck. We have current availability of the 9-valent. It is commercially available. We are confident in our supply of both the 9-valent as well as continued supply of Gardasil. We are also waiting for greater commercial coverage from an insurance perspective where it will be more broadly available in the U.S.
Thank you. Okay, Dr. Santoli for the VFC resolution.
Thank you. So the purpose of the resolution today is to update the resolution to include use of 9-valent HPV vaccine and to clarify the schedule and timing of vaccine intervals. As you see, the eligible group section is on the table. The additional column on the far right that's in yellow has been added to reflect the new 9-valent vaccine. The remainder of the table that's in white is the table as it currently exists in the resolution. We did use this opportunity to clarify the language in the schedule and interval section. It should be very familiar to you as much of it mirrors what's in the policy note. So the recommendation for routine vaccination at age 11 or 12 years, and then eligible females and males as young as 9 years old may be vaccinated. Vaccination is recommended for females and males 13 through 18 years of age who have not been previously vaccinated or who have not completed the full series. Then again, HPV 2, 4, and 9 are each administered in a 3-dose schedule. The second dose is administered at least 1 to 2 months after the first dose, and the third dose at least 6 months after the first dose. If vaccination providers do not know or do not have available the HPV vaccine product previously administered or in clinical settings transitioning to HPV-9 for protection against HPV 16 and 18, any available HPV vaccine product may be used to continue or complete the series for females. HPV-9 or HPV-4 may be used to continue or complete the series for males. Again, this mirrors the language you were just discussing. And then in terms of interrupted vaccination schedule and minimum intervals, if the vaccine schedule is interrupted, the vaccine series does not need to be restarted. The first and second doses should be separated by an interval of at least 4 weeks. The second and third doses should be separated by an interval of at least 12 weeks, with a minimum interval of 24 weeks between the first and third dose. The dosage information is referred to the package inserts, as are the contraindications and precautions. And then the statement about referring to published links once additional publications are made. And that concludes the update to the resolution.
Are there any questions? Yes, Dr. Harriman.
Just one quick comment on the intervals. Wouldn't it be valuable to perhaps use the exact same language that's used in the policy note in the VFC, rather than weeks and months and?
We can certainly do that.
And some of the minimal intervals historically have been in weeks, and that relates to something to do with the registries and how some of this is used. So they do mirror each other, let me say that. The minimal interval, I didn't show that in what we discussed because the minimal intervals haven't changed.
I think as a clarification, the forecasting for the systems need specific numbers of days, which you don't get with a month because months can be variable. And so as long as this is consistent with other VFC resolutions out there, I think we can let that stand. Dr. Herman?
Oh, that's fine. It's just nice if language is harmonized throughout. Maybe it needs to go the other direction. The policy note should mirror VFC or something. But
Dr. Shuckett?
The readers of the VFC note are not the same. So I mean, obviously, there's plenty of strengthening the quality of how we write these things that we'll take back.
Other questions or comments on this VFC resolution? And seeing none, let me ask see if there is a motion for Dr. Kemp. Any second. Dr. Romero. Any further discussion? And if not, we can start with Ms. Pellegrini and go to your right, which is me.
Pellegrini, yes.
Tempte, yes.
Bennett, yes.
Rubin, yes.
Romero, yes.
Harriman, yes.
Harrison, yes.
Belongia, yes.
Karen, yes.
Camp, yes.
Riley, yes.
Bocchini, yes.
Vasquez, yes.
Campus Outcall, yes.
Reingold, abstain.
And for the VFC resolution, we have 14-4, 1 abstention. The motion carries. As we're queuing up the slides for Japanese encephalitis, understand there's one more of the public comments who needs to leave. And I believe that is Carl Buer. If you can come up to the microphone and we can take your comment as we're getting ready for the session on Japanese encephalitis. Thank you so much for your flexibility.
Yellow fever, not Japanese encephalitis.
Thank you, guys. I'm tall, so hopefully this microphone works okay. As you said, I'm Carl Buer. I'm so grateful to have the chance to speak here today representing the National Meningitis Association. I survived bacterial meningitis when I was 14, and just last year learned it was serogroup B. I spent 5 months in the hospital, lost 3 of my fingers and both of my legs below the knee on Halloween. I had 11 surgeries. It took me almost 4 years to learn how to walk again on my prosthetic legs. To date, my medical bills have far exceeded $2 million. However, I still consider myself one of the lucky ones. 11 years later, I have a college degree, a great job, and a beautiful life. Most survivors I know feel the same way. We have pretty good attitudes and been able to find the positives in this disease. For example, my wife and I like to talk about my perks list, which includes things as my feet no longer falling asleep and the ability to know exactly what shoe size I need. However, there are also some cons. I can no longer enjoy fuzzy slippers, and Ziploc bags have become my worst enemy. But no matter how lucky we are, most of us will deal with the effects of this disease for the rest of our lives. My fellow survivor Samantha has had more than 30 surgeries in 37 years and still deals with pain on a daily basis. And there's Kyla, who lost both legs and most of her fingers and has gone through a dozen surgeries in the past 6 years to separate and lengthen her thumb, because, as she says, opposition is everything. Blake, who survived meningitis at age 18, learned so much as a dialysis patient before he got his transplant that he became a dialysis nurse. Some of us, like Casey and Francesca, have invisible scars like loss of cognitive function, hearing, or vision. Many of us endured, and some continue to endure, anxiety, fear, and depression because of this terrible disease. When we get together, we talk about the challenges that we deal with that most people don't even think about, like how to take a shower when you are traveling with no legs, or how hard it is to count to 10 on your fingers when you're missing a few. For most of us, it will affect our finances for the rest of our lives. Our cost of living will always be higher, since we may need additional support. There are the more obvious things, such as we may need more time off work to deal with medical complications or additional surgeries. Even those of us with great insurance plans pay an enormous amount in co-pays and out-of-pocket expenses. But there are other things that no insurance will cover, such as the cost to renovate homes so we can navigate in our wheelchairs or retrofitting cars to fit our needs. We are all very different, but there's one thing we universally agree on. We don't want anyone to have to go through what we went through and still deal with on a daily basis. When you look at the cost of prevention, I hope you'll calculate how high the cost of survival is as well. Thank you very much for your time.
Thank you very much, Carl, for your comments.